Dear viewers, this video is part 2 of the 4-part series on the WBCs, granulocytes, in the Medicine for Anyone in the YouTube channel. Lifespan of white blood cells. The life of the granulocytes after being released from the bone marrow is normally 4 to 8 hours circulating in the blood and another 4 to 5 days in tissues where they are needed. In times of serious tissue infection, this total lifespan is often shortened to only a few hours because the granulocytes proceed even more rapidly to the infected area, perform their functions, and in the process are themselves destroyed. The monocytes also have a short transit time, 10 to 20 hours in the blood, before wandering through the capillary membranes into the tissues. Once in the tissues, they swell to much larger sizes to become tissue macrophages, and in this form, they can live for months until destroyed while performing phagocytic functions. These tissue macrophages are the basis of the tissue macrophage system discussed in greater detail later, which provides continuing defense against infection. Lymphocytes enter the circulatory system continually, along with drainage of lymph from the lymph nodes and other lymphoid tissue. After a few hours, they pass out of the blood back into the tissues by diapedesis or extravasation. Then they re-enter the lymph and return to the blood again and again. Thus, there is continual circulation of lymphocytes through the body. Lymphocytes have lifespans of weeks or months, depending on the body's need for these cells. The platelets in the blood are replaced about once in every 10 days. In other words, about 30,000 platelets are formed each day for each microliter of blood. Dear listeners, by the way, this educational video is made possible by Prescribe-On, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribeon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Neutrophils and macrophages defend against infections. It is mainly the neutrophils and tissue macrophages that attack and destroy invading bacteria, viruses, and other harmful agents. The neutrophils are mature cells that can attack and destroy bacteria even in the circulating blood. Conversely, the tissue macrophages begin life as blood monocytes, which are immature cells while still in the blood and have little ability to fight infectious agents at that time. However, once they enter the tissues, they begin to swell, sometimes increasing their diameters as much as 5-fold to as great as 60 to 80 micrometers, a size that can barely be seen with the naked eye. These cells are now called macrophages, and they are extremely capable of combating disease agents in the tissues. This figure shows neutrophils moving through capillary walls by diapedesis and are attracted to the chemotactic force in the tissues. This figure shows the rolling neutrophils inside the capillary walls, named number 1, gets attached to capillary walls, named number 2, by adhesion molecules. Then, by diapedesis, named number 3, the neutrophil moves out to tissues where it is needed by migration, number 4. White blood cells enter the tissue spaces by diapedesis. Figure migration of neutrophils by diapedesis through capillary pores, the cell number 3. and by chemotaxis toward an area of tissue damage. See both figures. Neutrophils and monocytes can squeeze through gaps between endothelial cells of the blood capillaries and postcapillary venules by diapedesis. Although the intercellular gaps are much wider than a cell, a small portion of the cell slides through the gap at a time. The portion sliding through is momentarily constricted to the size of the gap as shown in figure earlier shown. White blood cells move through tissue spaces by amoeboid motion. Both neutrophils and macrophages can move through the tissues by amoeboid motion. Some cells move at velocities as great as 40 micrometer per minute, a distance as great as their own length each minute. White blood cells are attracted to inflamed tissue areas by chemotaxis. Many different chemical substances in the tissues cause both neutrophils and macrophages to move toward the source of the chemical. This phenomenon shown in figure, movement of neutrophils by diapedesis through capillary pores and by chemotaxis toward an area of tissue damage, is known as chemotaxis. When a tissue becomes inflamed, at least a dozen different products that can cause chemotaxis towards the inflamed area are formed. They include the following: 1. Some of the bacterial or viral toxins. 2. Degenerative products of the inflamed tissues. 3. Several reaction products of the complement complex activated in inflamed tissues. And 4. Several reaction products caused by plasma clotting in the inflamed area as well as other substances. As shown in the earlier figure, chemotaxis depends on the concentration gradient of the chemotactic substance. The concentration is greatest near the source, which directs the unidirectional movement of the WBCs. Chemotaxis is effective up to 100 micrometers away from an inflamed tissue. Therefore, because almost no tissue area is more than 50 micrometers away from a capillary, the chemotactic signal can easily move hordes of WBCs from the capillaries into the inflamed area. This figure shows the process of phagocytosis of pathogens. First, a microbe gets attached to antibodies and then gets engulfed by a cavity in the cellular outer membrane of the macrophage. The microbe then gets into the cell inside a phagosome, which merges with a lysosome to form a phagolysosome in which the microbe is digested. Thereafter, the microbial debris is extruded out of the cell. Phagocytosis. See figure phagocytosis of pathogens shown earlier. A major function of the neutrophils and macrophages is phagocytosis, which means cellular ingestion of the offending agent. Phagocytes must be selective of the material that is phagocytized. Otherwise, normal cells and structures of the body might be ingested. Whether phagocytosis will occur especially depends on three selective procedures. See the figure earlier. First, most natural structures in the tissues have smooth surfaces which resist phagocytosis. However, if the surface is rough, the likelihood of phagocytosis is increased. Second, most natural substances of the body have protective protein coats that repel the phagocytes. Conversely, most dead tissues and foreign particles have no protective coats, which makes them susceptible to phagocytosis. Third, the immune system of the body develops antibodies against infectious agents such as bacteria. The antibodies then adhere to the bacterial membranes and thereby make the bacteria especially susceptible to phagocytosis. To do this, the antibody molecule also combines with the C3 product of the complement cascade which is an additional part of the immune system. The C3 molecules in turn attach to receptors on the phagocyte membrane, thus initiating phagocytosis. This process whereby a pathogen is selected for phagocytosis and destruction is called opsonization. Phagocytosis by neutrophils. The neutrophils entering the tissues are already mature cells that can immediately begin phagocytosis. On approaching a particle to be phagocytosed, the neutrophil first attaches itself to the particle and then projects pseudopodia in all directions around the particle. The pseudopodia meet one another on the opposite side and fuse. This action creates an enclosed chamber that contains the phagocytized particle. Then, the chamber invaginates to the inside of the cytoplasmic cavity and breaks away from the outer cell membrane to form a free-floating phagocytic vesicle, also called a phagosome, inside the cytoplasm. A single neutrophil can usually phagocytose 3 to 20 bacteria before the neutrophil becomes inactivated and dies. Phagocytosis by macrophages. Macrophages are the end-stage product of monocytes that enter the tissues from the blood. When activated by the immune system, they are much more powerful phagocytes than neutrophils, often capable of phagocytosing as many as 100 bacteria. They also have the ability to engulf much larger particles, even whole RBCs, or occasionally malarial parasites, whereas neutrophils are not capable of phagocytosing particles much larger than bacteria. Also, after digesting particles, macrophages can extrude the residual products and often survive and function for many more months. Once phagocytosed, most particles are digested by intracellular enzymes. Once a foreign particle has been phagocytosed, the lysosomes and other cytoplasmic granules in the neutrophil or macrophage immediately come into contact with the phagocytic vesicle and their membranes fuse, thereby dumping many digestive enzymes and bactericidal agents into the vesicle. Thus, the phagocytic vesicle now becomes a digestive vesicle, and digestion of the phagocytosed particles begins immediately. Both neutrophils and macrophages contain an abundance of lysosomes filled with proteolytic enzymes, especially geared for digesting bacteria and other foreign protein matter. The lysosomes of macrophages, but not of neutrophils, also contain large amounts of lipases, which digest the thick lipid membranes possessed by some bacteria, such as the tuberculosis bacillus. Neutrophils and macrophages can kill bacteria. In addition to the digestion of ingested bacteria in phagosomes, neutrophils and macrophages contain bactericidal agents that kill most bacteria even when the lysosomal enzymes fail to digest them. This characteristic is especially important because some bacteria have protective coats or other factors that prevent their destruction by digestive enzymes. Much of the killing effect results from several powerful oxidizing agents formed by enzymes in the membrane of the phagosome or by a special organelle called the peroxisome. These oxidizing agents include large quantities of superoxide anion, hydrogen peroxide, and hydroxyl ions, which are lethal to most bacteria even in small quantities. Also, one of the lysosomal enzymes, myeloperoxidase, catalyzes the reaction between hydrogen peroxide and chloride ions to form hypochlorite, which is exceedingly bactericidal. Some bacteria, notably the tuberculosis bacillus, have coats that are resistant to lysosomal digestion and also secrete substances that partially resist the killing effects of the neutrophils and macrophages. These bacteria are responsible for many chronic diseases such as tuberculosis. Monocyte macrophage cell system, reticuloendothelial system. Macrophages are mobile cells capable of wandering through the tissues. However, after entering the tissues and becoming macrophages, another large portion of monocytes becomes attached to the tissues and remain attached for months or even years until they are called on to perform specific local protective functions. They have the same capabilities as the mobile macrophages to phagocytose large quantities of bacteria, viruses, necrotic tissue, or other foreign particles in the tissue. In addition, when appropriately stimulated, they can break away from their attachments and once again become mobile macrophages that respond to chemotaxis and all the other stimuli related to the inflammatory process. Thus, the body has a widespread monocyte macrophage system in virtually all tissue areas. The total combination of monocytes, mobile macrophages, fixed tissue macrophages, and a few specialized endothelial cells in the bone marrow, spleen, and lymph nodes is called the reticuloendothelial system. However, all or almost all these cells originate from monocytic stem cells. Therefore, the reticuloendothelial system is almost synonymous with the monocyte macrophage system. The term reticuloendothelial system is much better known in medical literature than the term monocyte macrophage system. The term reticuloendothelial system is a generalized phagocytic system located in all tissues, especially in the tissue areas where large quantities of particles, toxins, and other unwanted substances must be destroyed. Tissue macrophages in skin and subcutaneous tissues. Histiocytes. The skin is mainly impregnable to infectious agents except when it is broken. When infection begins in a subcutaneous tissue and local inflammation ensues, local tissue macrophages can divide in situ at that place or situation and form still more macrophages. Then they perform the usual functions of attacking and destroying the infectious agents as they normally do. Macrophages in lymph nodes. Essentially, no particulate matter that enters the tissues, such as bacteria, can be absorbed directly through the capillary membranes into the blood. Instead, if the particles are not destroyed locally in the tissues, they enter the lymph and flow to the lymph nodes located intermittently along the course of the lymph flow. The foreign particles are then trapped in these nodes in a meshwork of sinuses lined by tissue macrophages. This figure shows a section of lymph node showing the artery and the vein in the hilus, that is the entrance of the lymph node. The medulla, which is the inner portion, and the outer cortex are shown, as well as the germinal centers and the paracortical areas. The functional diagram of a lymph node shown earlier illustrates the general organization of the lymph node, showing the lymph entering through the lymph node capsule via afferent lymphatics, then flowing through the nodal medullary sinuses, and finally passing out the hilus into efferent lymphatics that eventually empty into the venous blood. Large numbers of macrophages line the lymph sinuses, and if any particles enter the sinuses by way of the lymph, the macrophages phagocytose them and prevent general dissemination throughout the body. Alveolar macrophages in lungs. Another route whereby invading organisms frequently enter the body is through the lungs. Large numbers of tissue macrophages are present as integral components of the alveolar walls. They can phagocytose particles that become entrapped in the alveoli. If the particles are digestible, the macrophages can also digest them and release the digestive products into the lymph. If the particle is not digestible, the macrophages often form a giant cell capsule around the particle until such time, if ever, that it can be slowly dissolved. Such capsules are frequently formed around tuberculosis bacilli, silica dust particles, and even carbon particles. Macrophages, that is, Kupffer cells in liver sinusoids. Another route whereby bacteria invade the body is through the gastrointestinal tract. Large numbers of bacteria from ingested food constantly pass through the gastrointestinal mucosa into the portal blood. This figure of a cut section of a small portion of the liver shows the liver sinusoids, which is spanned by many Kupffer cells. Figure Kupffer cells. Please review the same. Before this blood enters the general circulation, it passes through the liver sinusoids, which are lined with tissue macrophages called Kupffer cells, shown in figure Kupffer cells. These cells form such an effective particulate filtration system that almost none of the bacteria from the gastrointestinal tract pass from the portal blood into the general systemic circulation. Kupffer cells phagocytose a single bacterium in less than 0.01 second. Macrophages of spleen and bone marrow. If an invading organism succeeds in entering the general circulation, there are other lines of defense by the tissue macrophage system, especially by macrophages of the spleen and bone marrow. In both these tissues, macrophages become entrapped by the reticular mesher of the two organs, and when foreign particles come into contact with these macrophages, they are phagocytosed. So, dear viewers, this is the end of part 2 of the 4-part series on the WBCs, granulocytes, in the Medicine for Anyone in the YouTube channel. Thank you for watching the videos.